Hello, we shall discuss on the application of biotechnology in agriculture. Under this, we shall be discussing or focusing mostly on the genetically modified crop plants or transgenic plants. The plant is obtained by the transfer of gene or genes from some unrelated organisms through genetic engineering are referred to as transgenic plants, and so those transgenic plants are those plants which are having foreign genes in their genome. Transgenic plants are being produced because of the following objective. The first one is the pest resistance. Farmers use varieties of insecticides to protect their crops from insect infestation. But the use of these crop treated with pesticides poses potential health hazards. Therefore, in order to eliminate the use of chemical fertilizers, chemical insecticides and pesticides, pest resistance transgenic crop have been developed. Some of these transgenic crop that developed so far are BT cotton and nematode resistance varieties like potato, tomato, brinjal, etc. The second objective is for herbicides or weedicides tolerance. Manual removals of weeds from the crop field is expensive as well as very time consuming and tedious. Therefore, farmers spray large quantities of herbicides which may harm the crop plants and even pollute the environment. Hence, genetically modified crops have been developed such as cotton, maize, tobacco and even soybean in order to tolerate these herbicides and also biodegrade these herbicides. The third objective is for tolerance to abiotic stress. By introducing the genes from the wild relative of cropland that is growing in adverse environmental conditions into the domestically grown cropland, this cropland which are carrying the gene from the wild relative have been able to grow in the harsh environmental conditions and able to tolerate even frost, cold, heat, salinity or air or heavy metal toxicity. The fourth objective is for disease resistance. Genetically modified plants have been developed to resist the infectious diseases and are being developed to save our crop plants or forests from fungal, bacterial and viral attacks. Some of the viral resistant varieties of tobacco, rice, papaya, etc. have been developed so far. The fifth top objectives is the enhanced nutritional value of food. Malnutrition is a common problem in the third world countries, and in these countries rice and potato form the staple food for these people, but this rice and potato many a time they do not contain adequate nutrients. Therefore, recently golden rice has been developed, and this golden rice have been enriched with vitamin A, and also protein rich potato have been developed through genetic engineering to tackle the problem of malnutrition. The fifth objective, the sixth objective is the mineral utilizations. Crop plants have been modified for increased efficiency of mineral utilization. This conserves the soil fertility and reduces excessive additions of fertilizers. The seventh objective is for pharmaceuticals products. Transgenic plants are aimed at producing pharmaceuticals like insulins, vitamin A, blood clotting factors, etc. And cultivation of these transgenic plants for pharmaceutical purposes is called farming or molecular farming. Now we shall be discussing on some genetically modified plant. The first one will be BT cotton, or BT stand for Bacillus thuringiensis. Now BT cotton is a transgenic variety of cotton that has been genetically engineered by introducing a gene from the Bacillus thuringiensis bacteria, and this Bacillus thuringiensis bacteria produces an insecticidal protein that is toxic to the larvae of the insect of the order Lepidoptera, Coleoptera and Diphtheria. Now Bacillus thuringiensis is a soil bacterium, and it possesses a gene that produces a toxic protein that kills the larvae or even the adult insect, and these proteins they occur in the form of crystalline deposits in the bacterial spores, and therefore these proteins are also called as crystalline protein, or in short is called cry protein. So the gene that is coding for this crystalline protein or cry protein is called as crystalline gene or cry gene. Now the crystalline protein is not toxic to the bacterium itself, but it is because in bacteria this crystalline protein or a toxin exists in an inactive prototoxin form, but once this toxin enters into the midgut of the insect, this toxin become activated, and it causes swelling and lysis of the epithelial cell that is lying in the midgut of the insect, and this causes death to the larvae or even the adult. So this is all about the bt cotton that is being developed from the transfer of the genes from the bacillus thuringiensis. Now the second transgenic crop is the flavor saver tomato. This flavor saver is a transgenic variety of tomato and was engineered to have resistance to rotting using antisense technology. Rotting usually occurs due to the softening of the cell wall during the fruit ripening by the action of the enzyme polygalacturonase. Now, the ripening, fruit ripening can be delayed by blocking or slowing down the production of an enzyme that is polygalacturonase by RNA interference. RNA interference refers to the silencing the gene expression. Silencing here refers to blocking or delaying the gene expression. So the enzyme that is coding the gene that is coding for polygalacturonase can be silenced by RNA interference. So once this gene is being silenced, this enzyme that is polygalacturonase cannot be produced, and hence this will prolong the ripening of the fruits. And it is because of this reason that the fruits of the flavor saver tomato remains fresh and retain their flavor much longer. So flavor saver variety of tomato was the first commercially grown genetically engineered fruit. The third type of genetically modified crop is the golden rice. Golden rice is a genetically modified rice variety. The grain of this golden rice is yellow in color because they contain the pigment that is beta carotene, which is the precursor of vitamin A. And this has been developed by introducing the two genes from the enzyme phytoin synthetase that is being produced from daffodil plant, and one gene from the bacterium with the help of the agrobacterium. So these are some of the genetically modified crops.